Hello and welcome to another Micro Minute with me, Marty Jopson. Now, today I'm going to get straight into it because this one's a little bit complicated. We're going back to the plants. Oh, yes, this is for all the plant nerds out there, and you know who you are because this was a request. What I'm going to try and do is show you not only the top surface of this leaf here, but the bottom surface of this leaf underneath this microscope. It's all a little bit tricksy. Now, this plant, it's actually this plant here, which you can just about see. I took a picture of it earlier. Here it is. This plant it's called Maranta leuconura or something like that. The prayer plant. It's a pretty common house plant. It's one we've got here. And it's called a prayer plant because at night, apparently, the leaves kind of sort of fold down or something. Why that makes it a prayer plant, I do not know. But anyway, that's what it's called, and it's rather fun. So let's get on with this. What I'm going to do is I have my leaf here. I've got my little camera here. I'm going to prepare this specimen by cutting myself two little squares of leaf material. So we'll start with a nice clean, and you can see this was a manky leaf. So it's, I'm not doing anything horrible to the plant. And then I'll sort of cut there like that. And then we'll cut there. Oh, I'm not, get it in. And then I'm going to just go 1, 2, and then, right. So I've got two little squares of plant material, and I need to put my glasses on because I need to make sure I get one of them the right way up and one of them the wrong way up, so to speak. So, come back. Okay, which way around is that? I think that's the bottom. That's the top. Okay, so what am I going to do? I've got to get these the right way up. That's definitely the bottom. Right, I'll put a little bit of water on my slide. That always helps, not least because it helps stick everything down. And then I'll put that one on there like that. Ooh, it's floating about a bit. And that one on there like that. Okay, like that. And then I just realised I've forgotten something important. I have to rush over here. I can tell this is live. I come back here. I needed a tissue. And then I have another very thin piece of glass here called a Coverslip. And I pop that over the top there like that. Okay. And then I squash it all flat like that and squeeze out the excess. And what this does is this helps the microscopy, but it also helps stick everything down. Right, I think that should be all right. Now, I'm going to pop this under, so we'll lose the desk cam and come back onto the main camera. There we go, take the glasses off. This is going to go under here. I'm going to have a little look round and just to make sure everything's what I want it to be. Where are you? Focus, focus, focus. Is this the right bit? Yeah, this is the right bit. Okay. And focus. Boom, there we go. Right, this is good, this is good. So what I'm going to do is I'll find a nice bit, turn the brightness up a little bit. It's okay, it's not, I've had it better. I'm just going to squash that down a little bit more and go in there. Right, that's better, that's better. So, let's find a good section, and this is the trouble with this sort of live microscopy, is you've always got to find the best bit of the specimen to show you, which is the complicated and difficult bit for me. Right, here we go. This'll do, this'll do, this'll do. Let's flip that over and I'll show you what I can see. So here we go. And I'll put myself up in the corner there. And what you've got there is the top surface of the leaf. And you can see, well, it's kind of a sort of vague, bubbly kind of texture to it. Can you see that? It's sort of vaguely, that's about right there. So you can sort of see over here. Oh, actually, I don't want it to do that. I want it to do this. I want the pointer. Sort of over here, you've got these sort of bubbly bubblies, and these are the individual plant cells. And there's not a huge amount to see on the top. It's basically just a lot of green cells. So I'm going to go and show you the bottom of the leaf, though, which is more interesting. If I zoom over to the bottom of the leaf, this is better. This is what we wanted. Here we go. This is good. Look at this. And now suddenly you can see there's a little bit more going on there. Focus that. Because not only have you got the little bubbly cells, but you've also got these weird little kind of, I don't know, they almost look like eyes, or you might even call them little mouths, because these are what are known as stomata, singular stoma, several stomata, which means mouth, literally. And what these are, and this takes a little bit of explaining. So you have to think, what is a leaf doing? It's green because it does something called photosynthesis. And photosynthesis is where it takes water and sunlight and carbon dioxide gas, and that's really important, and turns it into sugars. That is the point of plants in a way, and it is the basis of all life on Earth, essentially. Now, the leaf is where that happens predominantly, but the leaf structure, it has a top surface and a bottom surface, but in between, there are all the cells doing the work, and they need to get the carbon dioxide. To get the carbon dioxide, there is a spongy hole in between the two surfaces of the leaf. And to get that carbon dioxide into that hole, you have to have little pores, little mouths on the undersurface of the leaf, usually, through which the carbon dioxide can go. Now, the way I like to think of this is bananas. Okay, because the cells that are lining this little hole, the little mouth, are banana shaped, so they're kind of that shaped. Okay, and that creates the hole through which the gas, the carbon dioxide gas, gets inside, so that it can go off and do the photosynthesis. And what happens, actually, is really clever, because what happens is at night time, when the photosynthesis stops, you shut all your stomata. The plant shuts its stomata to stop itself from drying out. And all it does is it straightens out its little banana cells. And as they straighten like that until they've gone completely straight, the gap disappears, and then they bend open again in the sun, and then they flatten out in things. So you could sort of open and shut the stomata. Right. So let's go back to my stomata. And there you can see them all individually. Now, these stomata, these are all currently shut. And what that means is that this plant is trying to conserve its water, probably, which probably means we need to give it a water. It's, after all, in a fairly warm environment here. Now, there is one other cool thing I wanted to show you, and that is I'm going to use some special optics in this to make these cells even more visible. Now, what I have in here, and I'm not going to explain it much at the moment because it's slightly complicated, is I have a way of using ultraviolet light. So this is invisible light, the stuff that you get from the sun that causes you burning. And I can illuminate the specimen with that rather than ordinary light. And when that happens, something rather funky happens. So have I got everything set up right? Yes, I have. All I have to do is flip that switch there, and pow, look at that! Suddenly we go from this sort of ordinary looking picture of a green leaf, and we end up with these amazing sort of weird alien landscape. And what you're seeing here is you're seeing parts of the leaf that are fluorescing. So in the same way that if you have a black light, an ultraviolet light, some things glow kind of, so they fluoresce, they kind of give off their own coloured light. It might be green or blue. And in this case, there's blue and there's a bit of red there. And you can suddenly see a lot more detail. First of all, you can see these things. Can you see these things here? And these things, these are trichomes. They're hairs, and I can sort of focus up and down on them. If I focus right to the end of them, there you go. Look at that. And they sort of show up this wonderful blue. Isn't that pretty? But if I focus back down onto the surface of the cell, now you can really easily see where all the different cells are. And if I use this device here, this allows me to zoom in. Can you see in that corner down there, there's a zoomed in little picture. And if I pick the right spot here, for example, no, that's not a good one there. That's the bottom of a cell. You can see the cells are kind of these wiggly wobbly shapes, basically just seeing the edges of the cells. And then, where is it? That is one of our stomata closed up. That's probably a better one just there. You can see a sort of a close-up of it, and it's closed up. But using this fluorescent technique, we can sort of see a lot more detail. So that's all I wanted to show you today. These are really quite small. The scale bar down here at the bottom corner there tells you how big they are. But they're super important for how leaves work. Now, I'll be doing another Micro Minute on Good Friday, this Friday, and I'm trying to think of something suitably Easter. Send me any ideas. I'm drawing a bit of a blank at the moment. It's quite difficult to find something that's interesting under a microscope that belongs in Easter. So thank you very much for watching. Retweet this, go to the website, and follow along and come along on Friday. Thanks very much. Bye-bye.